Thank you very much, and thank you for having me. Basically, the thrust of what I want to talk about today is our attempt to try to balance out the current imbalance between our ability to generate data and our ability to test the models that come from that data. I think we can all commiserate with the fact that we're generating these data sets at kind of an exponential rate. All of these various omics approaches and the literature that's online, as well as all the patient cohorts, are allowing us to generate hypotheses at a rate that far outstrips our ability to test them. Limitations in our models and tools and time are resulting in sort of a slowdown of this cycle that is intended to improve patient health and better treatment options. So there are a couple different ways that we can approach this, and one of them is really reflected in a lot of the NIH calls currently, and that's to better mine the data we have through computational methods and modeling. But the other approach we can use is actually to begin to improve our models and our tools and make them a little bit more high throughput. And so that's the whole idea behind our attempt to make primary cells a more genetically tractable model system. So the question that we attempted to ask is, can we make primary cells genetically tractable for the study of host-pathogen interactions, not needing to spend all of the time trying to use shRNAs and appropriate cell lines to try to validate model X, instead directly going into primary cells to look at some of these interactions would be a huge time saver. And the approach we decided to use to try this was using CRISPR-Cas9 ribonucleoproteins, or RNPs. Now, these especially don't need any introduction here at Berkeley, but just a quick one sentence. Basically, Cas9 is a programmable nuclease that will target a specific DNA sequence by complexing with a guide RNA. And the sequence of the guide RNA will dictate where this Cas9 nuclease cuts. And this has been behind a huge revolution in genome engineering in the last 5 years. And what was shown in 2015 by our collaborations in Alex Marson's lab at UCSF, together with Jennifer Doudna's lab, was that if you actually purify this guide RNA and Cas9 protein in vitro and then deliver them to primary cells by electroporation, you could achieve editing potentials up to 50% allelic efficiency, which was great and a huge breakthrough, but not quite high enough for us to begin applying this in a high throughput manner. And furthermore, the system they were using was fairly toxic. And so we collaborated with the Marson group and with Jennifer's group to try to revamp the system and generate a high throughput platform for genome editing in primary cells. And the cells I'm going to talk about today are T cells, and I'm going to focus on HIV, but we've now been applying it to a multitude of different primary cell types. So this is just a schematic of the platform that I'm going to be using today and talking about. So we take T cells from donor blood, isolate them out and activate them. You can activate them however you wish. These activated T cells are then nucleaffected with in vitro synthesized CRISPR-Cas9 RNPs. That generates a polyclonal knockout pool, which can be expanded with very little toxicity, used for validation by immunostaining or immunoblotting, and or at the DNA level by sequencing, and then these can be used for downstream phenotypic assays. So before jumping into some of the data, just a few notes on the platform and its capabilities. So right now we're editing about 100,000 to 1 million cells per reaction, though we're rapidly scaling this up in about a 20 microliter reaction, and are achieving efficiencies now consistently at a rate of about 95 to 99%. Each reaction only takes an hour and a half, and we're doing this in 96-well format using completely commercially available reagents, currently the Amaxa platform, with very low toxicity. It allows multiplexing, so you can make double, triple, even quadruple knockouts all at the same time, and it allows for freeze-thaw for ready storage at a cost of about $6.25 per reaction. So we really think we've finally developed a platform that allows us to begin doing high-throughput genome engineering in primary T cells. So just to take you through some of the data that we've been generating with this platform, some of the very first genes that we targeted as proof of principle were the HIV co-receptors, CXCR4 and CCR5. So for HIV to enter into a cell, it has to bind the CD4 receptor, and then either CCR5 or CXCR4 in a strain-specific manner. So first, just looking at some CCR5 data, here we're staining for CCR5 on the x-axis in unmodified cells, Cas9-only treated cells, or CCR5 knockout CRISPR RNP treated cells, and we can see that in those knockout cells we completely ablate CCR5 expression. And when we stain for CD25, which is a surrogate marker for T cell activation, versus CD4, we see absolutely no change in these other cell surface markers. That's quantified here in the green bars in the left, where we see an order of magnitude decrease in CCR5 levels. And then when we infect these cells with a CCR5 tropic HIV strain, we see an order of magnitude resistance to infectivity. And this can be rescued by pseudotyping with a CCR5 independent envelope like VSVG, indicating that the cells are not solely toxic, but that this is due to a specific editing event at the CCR5 locus. So we've now been using this platform, besides looking at genes that are already known, to begin to explore new data sets and new genes that people have been hypothesizing are involved in HIV replication. So we collaborated with Bruce Walker's lab at the Broad Institute, who had done a genome-wide CRISPR screen in a T-cell line, looking for genes that, upon repeated infection, provided resistance to HIV. And what they found was they found 5 genes in their screen which when knocked out, provided protection against infection. The receptor CD4, the co-receptor CCR5, and then 3 new genes that hadn't yet been described. And so we decided to take these genes and directly test them using our primary cell model. Now, 2 of these genes, TPSD2 and SLC35B2, actually reside in the same pathway, and that's the sulfonation pathway. So if something's going to be sulfated on the cell surface, it occurs in the Golgi apparatus. And so you have this sulfate donor, known as PAPS, which is transported into the Golgi by the action of SLC35B2. Then this sulfate can be donated to a target protein, such as CCR5, by the action of these TPST proteins, such as TPST2. So we took this and knocked it out in our primary cells and then stained for sulfated CCR5 on the Y versus total CCR5 on the X. And what we found in our non-targeting control is that most of the CCR5 on the cell surface actually is sulfated. But when we knock out SLC35B2, we can ablate most of that sulfonation. Now, when we take these populations and then infect them with a CCR5 tropic virus, what we see is that in blue is our CCR5 tropic virus. Unmodified in Cas9 only cells, we have high infectivity. When you knock out CCR5, you get an order of magnitude drop. And knocking out just the sulfate transporter itself phenocopies the receptor knockout. And this again can be rescued with VSVG pseudotyping across multiple donors, indicating that the sulfonation pathway is important in primary cell types for HIV infectivity. So that's still looking just at one gene at a time. So we wanted to see how high throughput we could make this. Could we do hundreds of genes at once from a single donor? And the data set that we decided to attempt to try was actually a protein-protein interaction data set that our lab had generated a few years back. So we had done a lot of protein-protein interaction work with HIV using APMS, identifying interactions in these nodes surrounding the yellow to each of the HIV proteins. So there are 438 interactors that we decided to go ahead and knock out in a couple of different donors, and then look at how these knockouts impacted HIV infectivity over time. So just how this platform looks is we arrayed 1,300 CRISPR RNAs in 96 well plates, complexed them with Cas9, electroporated them into a couple different donors, replica plated these, and then infected them with a GFP reporter NL43 strain of HIV. Every 3 days then, we took a time point and measured infectivity in each of these different wells. So on each of the plates, we had 6 controls. We had 3 non-targeting controls, which retained high infectivity across all of our plates. We knocked out CDK9, which is essential for HIV gene expression, and we saw consistent knockdown. We knocked out CCR4, which ablated infectivity in just about every plate and every sample, and then LEGF, which is a integration factor, which we knocked out and again saw consistency across our plates. So looking at all of the data together across all of our different guides, we see they all actually cluster tightly around this median, but we have a number of genes that either increase or decrease infectivity. So just pulling some of these out then, we can pull out the data at days 3, 5, and 7 during our spread. And what we can see, for example, here is knockout of CBF-beta, which is essential for the function of HIV-VIF. Without CBF-beta, VIF cannot degrade the antiviral A3 factor. And when we knock out CBF-beta in these primary cells, as shown in light blue or green here, we see a decrease from our non-targeting in gray relative to our other controls, LEGF in red, CDK9 in orange, or CXCR4 in black. And besides CBF-beta, we also hit elongin BC and elongin C here, and all of them showed a similar phenotype. And besides that, we're also finding new hits. So for example, here we're looking at knockout of 3 different factors. HUE1, which when we knock out increases HIV infectivity. SNU13, which decreases it, and UBR4. Now this is looking at everything in a gene-specific manner, but we can also look from the HIV perspective. So for example, here are all of the different guides targeting host factors that interact with HIV integrase. And when we array these, we see most of them do not have an impact on HIV infectivity when knocked out, but we do see some that increase or decrease it. And when we array them again then in our replication spreading profiles, we see our non-targeting guides cluster strongly right in the middle, and we see a lot of things that decrease HIV infectivity, our known controls, CDK9, LEGF, CXCR4, as well as other known factors such as gemin-2 and the importance. But we're also picking up new things such as FAM96B, which is a mitochondrial transport protein. And we also are knocking out things where we're seeing increases. So for example, INI1 or HRP2 and XRCC6. And what's cool about this data is that you can then go back and take our banked samples and probe to make sure that our knockout efficiency correlates with our infectivity phenotype. So for example, only XRCC6 guide 2 gave us a phenotype, and that's the only one that validated in our knockout. So far we've screened 300 genes, we have 100 left to go, but we've identified a number of genes. Anything underlined has already been reported, anything not is novel. And we're working with a number of different groups to validate all of these different interactions. And I would highly encourage you to talk to a couple graduate students in our lab, Paige Haas and Kelsey Haas, who have posters here today talking about different CRISPR screens and technologies that we're using. And besides doing this, we're also expanding this to look at HIV latency, look at epistasis. We've expanded it to tissue cell types, macrophages, dendritic cells, and we're also now beginning to do high-throughput knock-ins. So with that, I would like to thank everybody in the Krogan lab, particularly a fantastic technician who's been working with me, Michael McGregor, as well as Kelsey and Paige Haas, who have posters here today. And then our fantastic collaborators, Alex Marson, a graduate student, Joseph Hyatt, who helped me do way too many plates of flow in the last month. Jennifer Doudna, of course, Viviana Simon at Mount Sinai, Jeff Cox here, and then Bruce Walker at the Regan. And with that, I'm happy to take any questions.
I'll start. I have a question. What's that third protein? You highlighted 3 new proteins, 2 that were involved in the sulfonated or the sulfate pathway, and what is the third one?
Yeah, so the third one was ALCAM, but they did their screen in Jurkat cells. And ALCAM isn't actually expressed on the surface of primary T cells, which speaks to sort of the trick of using these cell lines as your primary model. However, they were able to identify a different factor, ICAM, which is expressed highly on primary T cells, and when they knock that out, it decreases cell-to-cell transfer. So we're thinking that's probably why they picked it up in their screen.
Cool talk. I wanted to see how you control for cell proliferation in your 4 days of your screen.
Yeah, so we have hit a number of things which are lethal. And luckily, we're doing all of this as a flow base, so we have single cell information. And so prior to screening any of our data, we actually set cutoffs based on cell viability, which we gate versus by flow, but then also cell count. And usually, I mean, the knockouts are so efficient that if you do knock out something essential, the cells are just gone.
What do you know about the turnover of the RNP?
The what?
The RNP, your Cas9 CRISPR complex.
Yeah, what about it?
What about the turnover? I mean, how stable, how long does it persist in the cell? How stable is it?
We originally thought it only persisted in the cell for about 24 hours. And that was just based on flow staining. But then when we actually got a little more sensitive microscope, we found that actually they form very specific loci in the nucleus that last many days. So we don't know if that's the exact site of cutting. That's what we predict, because we're getting efficiencies way higher than you would predict just based on a single cut. So what we're thinking is that it actually localizes there and then cuts maybe multiple times until that site is destroyed. But yeah, that's all unknown still.
You mentioned you're going to start looking at different cell types. Within the T cells, were those naive memory, different populations of memory?
Yeah, so we've done different populations of memory, and we've done CD4 and CD8, and we've done tissue-derived and peripheral. We're still working to get monocytes working robustly. They're proving to be a little more tricky. But the other thing we're really interested in doing is getting resting T cells to work well. And what we found is we're able to hit very specific genes in resting T cells with just as high efficiency as we are in activated T cells. And what we think it has to do with is the chromatin architecture. And so I think some of these loci are going to be just so tightly bound, especially in a resting state, that we're not going to be able to target them efficiently without some sort of low-level stimulation.
What about, have you looked in these CD34 reservoir cells that are being reported?
I would love to, but no, we have not yet. It would be great to find a source to begin doing those with, but so far, no.
Okay, thank you. I think we'll stop here.